Hello. Thank you very much for being here. Here I will introduce for this evolutionary medicine meeting, and I am very glad to be here and to organize the symposium in which we will review these 3 talks and also have a roundtable. But here I want to introduce a bit the field is in a way not really new, but some years ago we are developing this kind of subfield of evolutionary medicine, or perhaps a subfield of EVO-DEVO, or both of them together, right? So what is evolutionary developmental pathology, or macroevolutionary medicine, as some people call it? Because it does, contrary a bit to evolutionary medicine, that tends to be more micro. Here is really, as you can see already in this picture, more macro, right? The bigger changes, so-called macro mutations and things like that, and to see if there are patterns or not in them, for instance. So how can we summarize this in a simple way to start with? Well, in evolutionary developmental pathology, or EVO-DEVO-PATH, there are 4 main parts. So one will be the comparative embryology, anatomy, more typical one, right? Then you have the developmental studies and genetical studies, using, for instance, model organisms like salamanders, mice, chickens, zebrafish, and so on. All these have been done by people that work with us or that collaborate with our labs. Now in my lab, I don't do any more like this type of research with animals. I don't think it's just ethical, but we have done this, or sometimes we have colleagues that can do that. But basically, we actually don't need so much, because what is changing also is that we are using humans as a main case study. How do we do that? We know a lot about human fossils, evolution, and so on, and also the cases of birth defects. So that no body is being damaged, we are using bodies of child that were dead since decades ago, or people that actually have birth defects, that we are working with surgeons and so on, right? So it's actually, that's the difference. Humans are the main case study for this subfield, and humans are the main case study that show about the evolution and even the development and pathologies and variations of other animals. This also coincides with our lab, that is, well, the Free Diogo lab, that is aimed to address broader medical and social implications. As you can see here, we talk about racism, machismo, etc., always based on empirical evidence, and to discuss the uses, abuses, and myths concerning these topics. So these are some contributions. For instance, we also try to find and to report untold stories about the contributions of, let's say, Muslims for the understanding of human anatomy, or about minorities for the understanding of human medicine, and so on. The base was really the first PhD I did. Actually, it was with catfish. Then I began to do other fishes, and then reptiles, amphibians, and so on, and mammals. And then actually I was invited to do a second PhD in anthropology, doing humans and other primates, dissections, and so on. But here I began to do the anthropology, talking more about birth defects, variations, and so on. And then the 2 worlds together combined did really what is now the EVO-DEVO-PATH. This book we did with about the evolution, development, and the homologies of all main chordates. So it's really like a broad approach, and that also allows us to see more a macro evolution and to relate with pathologies. That I think is what is kind of new about this talk and about this symposium, right? So just to summarize, for instance, we could also see mosaic evolution, and for instance in humans is a good case that it's so easy to understand, right? So this is the book that I wrote with Bernard Wood about the results of that PhD. I will of course not compile it here. It's more than 1,000 pages. But just as a summary that will be important for our for this talk, is that, for instance, humans have more muscles in some regions of the body than most other animals. But in the rest of the body, we are actually very simplified animals. We have less muscles in our body in total than, for instance, a mouse. So we have to really understand that. So that is really like a mosaic evolution. interesting also for the topic of this talk is that when you talk about evolutionary medicine, one tends to be very adaptationist. That is something that some of us in the field are trying to change, and lately it's changing a bit. But majority of evolutionary medicine, as for instance, evolutionary psychology, behavioral ecology, they are still very much adaptationists, like finding a reason that is adaptive for almost everything. As some people even begin to make fun of themselves when they had this idea some decades ago, right? That I know even suicide or even mental pathologies or overdoses, they will always be adaptive for something, as if other animals, or there is a mismatch only, right? As if there are other animals are not sick also, or don't have. Of course, mismatches are important in human evolution, but that's really a wrong concept to only think about mismatch when other animals also suffer from a lot of pathologies and so on, and many of them are just a byproduct. And this type of view of Stephen Jay Gould in the 70s and the 80s did not really permeate so much this field so far, what is interesting. I think the reason is that many evolutionary psychologists, evolutionary people doing evolutionary medicine are not really people that did so much evolution. They came from other fields, obviously from psychology, from medicine and so on. And so sometimes they use these more old dated. But this graph is actually interesting to show these trends, because Stephen Jay Gould actually said that for this no trend does not mean that something is wrong. For instance, in this case, is just to simplify, humans are the primate that evolved more in terms of muscles and so on, evolved more steps since the first primate, as we have measured in previous phylogenetic studies. But as you can see here, humans have less muscles and less bones than most primates. So changing more, but actually losing less. We're not getting more progressive or more complex anatomically and so on. Again, we are more simplified. So there is no progress, no trends of this kind. So really now talking more about EVO-DEVO-PATH. One of the main examples was a paper we published in Nature with a lot of colleagues that shows the amazing links between the head muscles and the heart muscles. But not only that they are connected somehow evolutionarily developmentally, they are amazingly connected in a way that a picture says more than 1,000 words. You have here, for instance, these muscles of the right side of the face that are embryologically and developmentally more strongly related directly with part of the heart than with the muscles, the facial muscles of the other side. It's amazing. Those ones will be more connected with this part of the heart and so on. As you can see, this is really EVO-DEVO-PATH, because we did studies from humans with birth defects. There are studies, developmental studies, genetic studies about this field, what we call the cardiopharyngeal field, developing really each gene changing gene expression to really form all these muscles and so on in model organisms. Broader comparison between chordates. And then what is the gain of that for evolutionary medicine? For instance, wow, in this case, there is a lot. That's not a coincidence that the paper was published in Nature, right? Because you can say to a doctor, well, we'll do ultrasound to a baby. It's very difficult to really see inside organs. So normally we try to detect birth defects from the superficial tissues and so on. But if this correlation is really existing also in human medicine, and we are doing now some studies to try to confirm that together with other colleagues, then you can, if you see, like, say, a baby with a problem in the ultrasound, a fetus, with problems already showing on the right facial muscles, let's say, the doctor will probably say, oh, now I know that I will really have to check as much as I can this part of the heart, because it can also be messed up, right? So that is the kind of implications that this has. Another implications actually is a book we wrote, and is already being used by some universities around the globe. It's that we can understand better, not memorize like in medical schools, and I teach a medical school and there are mnemonics and so on, and I don't think is the way to do it. It's better to try to understand why the human body is as it is, why evolutionarily and developmentally Because there are many things we can gain about that, not only to understand and to be doctors, but also to teach others, right? For instance, the true anatomical position that, as you know, probably since Da Vinci is being taught in a wrong way. The anatomical position that we teach is developmentally completely wrong. How you compare with other animals, you see the pathologies, you see the correlations, and of course, like this is the real drawing, like this is what is truly dorsal, right? And this is what is truly ventral. What does it mean? Because of the rotations of the upper and lower limb, that's why it seems kind of a mismatch. And so the front part of the leg, those muscles there, the skin and so on, correspond here to the back of the arm. So this is the kind of things that we really need to understand and eventually to change, but has to be done completely together in the world. So all the manuals, all the educational books and the exams and so on will combine it, right? Second, amazingly, and I think more important, it allows us to understand the imperfections of the human body. But it means imperfections in more like a theological way. It's just things that will make no sense if an engineer will do them. For instance, just one example among literally thousands of them in our body. The muscles here on the back of the shoulder, these muscles are limb muscles, are upper limb muscles. They are innervated by ventral rami, meaning rami that are on this side. So the nerves have to come from this side and go in strange ways to innervate muscles that are innervated by them, but on this side. It makes no sense. If it was like an engineer, of course, you will do the nerves going from the dorsal rami. But why is that the case? Because of evolution. We have to understand evolution. In fish, the muscles are mainly ventral of the pectoral fins. Then in evolution and in embryology of humans and other mammals, they migrate dorsally and take the nerves with them. And so the nerves can be really using very strange paths that can be very dangerous, like the axillary nerve goes here, and is really not the best thing you have to have an axillary nerve going on the axillary region and being so prone to be damaged. And actually, the most damaged nerve when you have injuries of the proximal upper limb is the axillary nerve. Right, so this is really like how to understand human anatomy and human pathologies. Also to understand and to emphasize that evolution is not just one thing. There are variations. There are also birth defects, a lot of them. There are pathologies, right? And those things exist. We know that almost in your body, at least 20 in average, 20 muscle variations will occur in every human body. Those are variations, but they occur in about 20 muscles in average. So there are a lot of them, but we try to minimize them. We don't talk so much about them, even if we know that variation is the mother of evolution, right? Examples, for instance, there is a palmaris longus, a muscle. When you do this movement, you will see like most people, 85%, will have two tendons here, but 15% of them don't have one of the tendons, the palmaris longus. Why? Because in other primates, that muscle is really important for the flexion, and you depend on your life if you are like a gibbon in that position. Because if you go down, perhaps there is a leopard or a lion to eat you. But in humans, we don't depend on being on trees, flexing the hand, and so on. So we are losing slowly, millions of years, the palmaris longus. It's called a polymorphism, not a variation, because it's present in more than 2% of the people, but is also not present in the majority of people. So that will be the normal phenotype, and polymorphism from 49 to 2%, and then variation. Variation, some of them are amazingly similar to things that we have lost in evolution in adults. We have lost in adults, and for some reason, some people have them until they are adults, and I will talk more about that. But some of them are very similar. Why? Because it makes sense. Most variations, some birth defects can be really strange, but most variations normally are things that were in the past present in our ancestors. They were almost never lost. They are in the embryo, they are in the fetus, but then disappear, but they can persist in some rare cases, variations, less than 2% in adults. This also has to do with the phenotypic stage. That's where most of the things happen and are very sensitive in embryology, the first weeks of formation. But very importantly, in those stages, as you will see, we have a lot of structures that are actually atavistic and will then disappear normally after. But if there was a huge mess up with the phenotypic stage, then some of these structures can persist until adult stages. Also, as you know, the phenotypic stage is so sensitive that is almost impossible to break some of the rules. And this is one thing we will talk more and more about evolutionary, about the pathology, the constraints. Again, against the adaptation theory that basically you can do whatever you want, the more constrained view of evolution of Stephen Jay Gould, right? That is really difficult in general to change, especially because of the phenotypic stage. I don't know if you know a work by Francis Gall, is a brilliant work, was about why most mammals, with only 2 or 3 exceptions, have 7 vertebrae, including giraffes that could have a huge neck. They still have 7 vertebrae. So why? And what she did, she went to hospitals, she saw correlations, and she saw that many babies that die with cancer and so on, they have reports or even their bodies are in museums and so on. They normally have more or less than 7 cervical vertebrae. Of course, they don't die because of that. They die because that is one of the many things pleiotropic together that go wrong during the phenotypic stage. If you mess up with that stage, many things can go wrong. The vertebrae are just showing that that phase is going wrong, and many other things, the ones that can lead to cancer, that is what killed most of those kids, also go wrong. The first implication of doing this kind of observations is understanding the true nature of the human body. So we can really say the homologies, where do these muscles come from? Well, they come from these muscles in fish and so on, right? So this is really important. And that's what we do in that book, right? We do by groups of real origin, right? Not the functional way that some doctors normally talk, you know, the rotator cuff, putting together 3 or 4 muscles that have nothing to do with each other embryologically in terms of innervation and so on. We group instead the muscles by innervation, by true embryological and evolutionary group. Now, I will give some case studies of things that we have done in the last 20 years in the lab, and that can be very diverse because, as you will see, we don't use a single animal. We don't even use animals anymore. We don't follow a single question. We try to be as broad as possible. In a way, it's trying to go a bit to do wonders of nature. But of course, like in a more informed way, we are not saying that we want to go back before the revolution in science and everything. But as this book, I really recommend this book, said, also is said to have lost this wonder for nature in this broader moralistic way of seeing things. And I agree with that part. In fact, what the EVO-DEVO-PATH is a bit similar to some of the things that much before that moment, Etienne Geoffroy-Saint-Hilaire talked about, you know, this view, this broader view of evolution together with birth defects and so on. Now, how can you do that, right? You need a lot of birth defects. And again, we don't use animals that are manipulated or made to suffer. So we use natural examples. The world is so diverse that you can use natural examples or natural mutants, as people call them, right? For instance, I especially deal with people that also want these wonders of life. I don't know if you know about Drew Norton or Howard Evans. If you go to their labs, it's like a collection of rarities, of these old museums of wonder. And for reasons I don't even know yet, Drew Norton gave me his collection before he retired. He's now traveling a lot in the world, and he's a good friend, and he gave me all these different specimens and so on. Many of them also given by Howard Evans. He's also still alive, emeritus professor at Cornell. And for instance, here is a study we did with cyclopic lambs of all types, right? And actually it's natural because it's the lambs have a lot of cyclopia because of types of vegetables they eat naturally. And so they gave me this collection. You can see here like the more normal lamb and in different stages until the cyclopia. And we dissected them and took scans of them and so on. For this is just for you to know, and I will begin to introduce you to the most important part, the most common pattern that we can see here, is that those lambs are very similar. The cyclopia in the lamb is very similar to the cyclopia that happens in fetuses or human babies. So it seems already, and I don't like to talk about this name in the sense that it's an ill name, but Pearl Arbor did it to simplify, right? The logic of monsters. Of course, not even the lambs or people, much less, also are monsters. They are no monsters. It's a social construction. But it's a way to say that even when there are huge developmental pathologies, there are still some logic, the constraints of development, the constraints of evolution. This is very important, you know, because in medicine, we still have this view that is changing slowly, that many birth defects like cyclopia, having only one eye, or anencephaly, that's not really like not having a head, but mainly almost lacking almost a brain, that everything is just a mess, that there is no, like, you know, there is no homeostasis, right? There is actually a theory that is called the lack of homeostasis, but that is really not true. We can see that, and I will show you some examples. There is really, even in those very severe cases, because that's what Per Arbor said. He said, even when evolution goes amazingly badly, you still have these logics. And that's why actually many defects in humans correspond to things that actually happen in evolution, in natural, normal evolution of other animals, or variations in humans happen. It's a mirror. There is a triangle with birth defects, variations, and evolution that is always paralleling each other and repeating. Why? Because there are not so many solutions again in nature. In contrast to the adaptationism, evolution is very constrained. All mammals have only, all tetrapods have only 4 limbs. You could be a better elephant, a more optimal elephant, perhaps with 8 limbs, or a whale, perhaps with 20. But still, the maximum is always 4 limbs. And even whales that don't really need hind limbs, they still might have them when they are embryos, and even sometimes when they are adults, small ones. So it's very constrained. And so, apart from the collections we got from other places, we also have collections like this in the lab, really hardcore birth defects that came from mothers that had these kids, gave 2 births and they were dead, and they gave them to Howard Hospital. And we did in the past, again, studies with model organisms. So just very quickly to talk, when you talk about even birth defects, much less even, or much worse, logic of monsters and so on, there are no monsters, and there is not even normal. Normal is a social construction. All these cases are real cases. We can be born like that. Not really like a single series, because there are 2 different pathologies here, but you can see that we can be very differently, right? And what is important is that right now I can say, oh, this is normal. And perhaps you say, no, this is normal for me. There is no normal. Normally, evolution is a continuum. Not always there are discrete characters, but mainly is a continuum. What we can say, the only thing here is that right now, these 2 phenotypes are the ones that most commonly arrive to the adult stage in humans right now, at this moment, at this place, right? That is the only thing. There is no normal or abnormal. So when I say normal, you understand that I'm simplifying. So we study a lot trisomies. As you know, there are many types of trisomies, even each chromosome, but in humans, the ones that normally can survive after birth are 13, 18, and 21. 13 and 18 are very severe. Normally they die the first year or second year. But 21, trisomy 21, Down syndrome, it can persist people until adulthood, as you know. We did books already about these subjects. So again, we had these fetuses or newborns. For instance, in this case, it was a newborn. a fetus with a trisomy cycropia, because the trisomies don't have always the same defects or pathologies. There are many types of pathologies that can happen in different fetuses with the same trisomy. It's very complex. But for instance, in this case, you can see the skeleton and also the muscles that we dissected, and then the 3D reconstructions and etc. by Peter Smith that worked in our lab, and we did this book about EVO-DEVO and EVO-DEVO-PATH of trisomy. We compare with normal, again, it's not normal, it's what is more common nowadays, newborns and fetuses, human newborns and fetuses. I will just give you an example. What can you learn from these things? For instance, we go another step. And apart from doing that, we do anatomical networks. It's a new method that can be applied to anatomy or to anything else, to use mathematical models, to use this holistic view of life. Because again, the problem with reductionism was reducing the whole to the parts. And that is not the reality. We now know that there is emergence. The total is more than the parts. So now new networks are really in vogue because it's precisely to study the whole and not just the parts. So here you see the networks and the modules here of the cranium of adult humans, here of the muscles, just the muscles. So you see in the muscles there are only 3 modules that really are strongly correlated with each other and internally and more far away from others that are just small. And amazingly, the 3 models is very interesting. They correspond here, this one, to the 2 sides of the facial muscles of the human head on the 2 sides. And then here you have kind of semi-modules. And this is really amazing because we did this, and a posteriori only, we saw that actually is really confirmed by medical studies. For instance, the paralysis on the mid-face is normally on one side. By anatomical studies that study the nerves that go to the muscles of right and left side of the upper face, they are much more similar to each other than the ones that go to 2 sides of the mid and lower face. That seems again to show that these are more semi-independent. And also correlates with human evolution. The asymmetric movements are actually the ones that normally show us more complex emotions like loving someone or missing someone and so on. This is like, of course, the more real network is everything together, bones and muscles, and seeing the modules we obtain. So now another level, we do the anatomical networks of so-called normal newborn heads, right? So here the cranium, skeleton, here the muscles only, and here the bones and the muscles. So just remember, so far we have seen, and it's logical, that the newborn has the same 3 modules of the facial muscles, right, than the adult, because the baby from day one needs to express and communicate with the mother and the father, and so that will have to be done, right? But interestingly, when you do, even to go to another level, to do to study birth defects as anatomical networks, amazingly, you see the constraints here. You begin to see the so-called logic of monsters. Look at this. Even if these fetuses die inside the mother, because supposedly everything is going like completely randomly, it's not going randomly, because even with one eye only, the 3 modules that we find in the normal newborn and normal adult head are still here. That is the logic of monsters. And again, these are not monsters. We will try some not monsters, but just using that name that was created by Per Harvard. Here is also interesting because we did just anatomical, because again, EVO-DEVO-PATH needs always a broader comparison, right? And so we did the anatomical networks with the normal adult head of mammals and their sister groups. Rats, rhesus, flying lemurs, and so on. And very interestingly, here on all these non-anthropoid, including tarsus, all these non-anthropoid primates and other groups, you can see that all the modules at the network modules are always very symmetrical, completely symmetrical. Now, when you arrive to anthropoids, meaning monkeys and apes, you start to see something very strange. You begin to have a single module, facial module, right, from anatomical networks that is asymmetric. And we know, as I said, that asymmetry is very important for to show complex emotions. And really, it's amazing because we're not expecting this, but humans are the ones that the left and right asymmetric modules have a higher proportion of all facial muscles of the head. So it's getting more and more asymmetric, not in terms of real anatomy. Really, real anatomy is symmetrical, mainly the facial muscles, right? I'm saying in terms of the modules of the networks that go a bit deeper to that, to see that there is the connections between them change and they're connected in a more asymmetrical way. That's basically what we can say. Now, I said already before that it's crucially important not only to see variations, but also so-called burst defects in people with trisomy and so on, to see also the constraints and, importantly, the uses for medicine. Again, macroevolutionary medicine, what macro changes can inform us. For instance, one amazing thing is that people with trisomy, not only 18 and 20 and 13, but even 21, people that go until adulthood and we dissected, well, after they died and so on, they tend to have some muscles that disappear millions of years ago in our adult ancestors. For instance, the platysma cervicale, as you probably, if you know a bit about anatomy, this is the only one platysma myoides that we now have normally adult humans. This muscle is present in almost all mammals, but disappear in adults, again, in adults since we have gorillas, chimpanzees, and humans. So more than 8-9 million years ago, our ancestor adults did not have this muscle. But for some reason, some humans have this muscle after birth or even until adult age, if they are trisomic 21 and so on. So this is a true atavism, right? Atavism is a muscle that was present in our ancestors that disappear in adults and then reappear in some adults. Now, amazingly, perhaps now you expect already, this muscle is present in all of us when we are embryos. You can see here, this was an older study, and then we also have more recent studies, as I will show you. We have this muscle when we are embryos, but normally it disappears very early when we are inside our mothers. But for some reason, it does not disappear in people, for instance, with birth defects, with trisomy, and so on. Same thing applies with some hand muscles that were present for a long time, disappeared in adults for like 20 million years ago, and suddenly are present in some people with trisomy and so on, adults, but also present in almost all the adults of chimpanzees. So it's a reversion, and you can now understand why do we have these reversions in nature. Well, not difficult, because again, these muscles are present in all human embryos. All of us had these muscles, but normally we lose them very, very early when we are like tiny, 3.5 centimeter embryos. But some people do not lose them. And in chimpanzees, probably the same happened. And those people actually were selected, and now all chimpanzees, or almost all, have these muscles in adults again. So reversion is actually not difficult to understand by understanding that the muscles were never really lost in evolution. They were lost in the adults only. They persist in the embryos as the potential. This also shows the potential, right, of evolution. But that potential is not because evolution, and that is the problem of adaptationism also in a way, right? Not because of evolution is predicting, oh, I need that potential, so I will keep it. No, it's mainly, the main reason clearly seems to be, and you can really see this here, because of the constraints, as Stefan Jay Gould said, right? First of all, there is no function. For people that will defend that there is function of everything at every time, if not, things will just disappear in nature, is not true at all. We can see this case especially here. There is clearly no function for a facial expression muscle of an embryo, right? If Before, we could not do ultrasounds. Nobody will see this expression, facial expression of an embryo inside the mother. So this is completely useless. But as you can see in this picture more clearly, why they are there? Because it's not so easy in evolution to say, look, I don't need any more. So these muscles, for instance, we don't have muscles adductores going to these digits here, but we have one still going to the thumb. It's very difficult in development saying, look, these ones just completely disappear from scratch. They don't even appear. I just have this one. That's the only one I need. No, that's not so easy, because you do have to form the contrahentis layer. And then, yes, slowly they can disappear, and only this one that will go to the thumb persists. But that's, you cannot just delete 4 of them and just have 1 of them. You have to form the whole layer, the mammalian layer, as people call them, and then specialize. That actually was Van Bauer's law, right? Now, the mechanisms, because you can have here and then we can stop here, right? But other colleagues, I don't do again experiments with animals, but other colleagues then can do experiments. For instance, they can have model mice for Down syndrome. And what it seems to be happening, why people with Down syndrome, for instance, will have these muscles more often, not always, but 30%, 40% more often than people without trisomies. Because it actually seems to be a gene that is located in a Down syndrome in mitochondrial models, so in humans also, that makes a way that there is that gene, right, is related to having less apoptosis. So what some of my colleagues think is that because people with Down syndrome, they have 3 copies and not only 2, right? 3 chromosomes, 21, not only 2. They have even less apoptosis, right? They have less apoptosis than normal. What is less apoptosis? Less cell death. And so these muscles that should die, right, during normal development, early stages, do not die until much later stages or even adulthood. And this seems to combine with one of the main causes of death of people with Down syndrome. I don't know if you know, but it's problems in the heart. The heart does not form the septa, etc., with apoptosis as it should. And that is also the main reason of death of many of these mice. So it does seem to be there. And you can see here the mechanisms that are so important to really understand this and apply it in a medical way. So I don't know if you have seen this. This came everywhere in the press, but some French team did the first study of human embryos with antibody staining. These are the things we have done for 20 years in other animals, but for some reason, I don't know if religious, and that's not a coincidence that this is done in France, but other reasons that some said these embryos are dead. These human embryos, of course, are dead. They just use them antibody to stain them as they use with embryos of other animals. But this just happened about 3 years ago. We never saw humans in this way. Probably for most of you, this is the first time you are seeing this a human embryo, 9 gestation weeks, seen from behind. So these are the back, the back muscles and so on. This is the head. This is the upper limb on the left side. And so I was invited to study these images, right? So to put these names and to study and to compare them with the development of other animals and so on, right? And there are amazing things that came from this study. So they came, of course, in the press because we did not know this about human embryos. There were some studies, as I show you, that show that we had some muscles that disappear later, yes, but not shown in this way and at this level of detail. For instance, here they are talking about the dorsometacarpalis, right? Is a muscle that we saw here. These muscles, dorsometacarpalis, are present in human embryos. Well, the dorsometacarpalis disappeared in adults, because clearly they don't disappear in embryos, disappeared in adults in the transition that led to mammals. We are talking about 250 million years that it disappeared in adults, and we still have them in embryos. You can see how constrained this is. But that is not the only thing. In our hand, we have about 21 muscles when we are adults. 21, 20 muscles, depending on people, accept more than others. Human embryos can have more than 40, more than double. So most of our muscles disappear during embryology. Very different from the idea that things are just dividing, and we have more and more. This old idea of progress, even in development, is completely wrong. Factually, developmentally, we lose a lot of muscles during development, vestigial muscles mainly, right? Atavistic muscles. An example that I will give also is that also has to do with the logic of monsters, but is really applied also to medicine and shows something that was really also very new in the field of evolutionary developmental biology in general and for medicine, right? When I was doing studies of salamanders, and I will explain why I was doing them, I realized for the salamanders have no digit 5, right? No digit 5. But the muscles of digit that normally go to digit 5 with all the tetrapods with 5 digits were still present there. But now they were going to digit 4. As if, strangely, the muscles don't know which digit is there. They just go to the more extreme. So in this case, more ulnar, in this case, more radial digits, right? Like if they were kind of blind, like those kids that put kind of a mask and don't see and just touch what is more close, near to them. But this was just like a guess. But then we begin to see with other my students and the lab and so on, and we published this paper to see, wow, this happens a lot. Happens the same with frogs. They have no digit 1, but they have muscles of digit 1 going to digit 2. Happens in birds and etc. And then again, we use the model, and the model is humans, birth defects, natural mutants, right? And so what we saw amazingly in a single newborn, but of course now we have a lot of examples. The trisomic 18, a different one from the one I showed before, this is newborn, that was really, really defective in the sense that it died after it was born. And one hand has 6 digits with a hexosum, and the other hand of the same newborn has 4 digits with no sum. But all the sum muscles with all the details in the bundles were there, but now they go to digit 2. Now to simplify, what can this mean? Well, there are 3 information types that you can talk about 2 digits. You have the developmental anlagen, saying, for instance, in birds, we know that for instance, that development and the identity can be different. So one thing is the homeotic or genetic identity. Another one is a development anlagen, and the other one is just topological position again, being more radial, more ulnar. What our studies really show, comparative studies, is that the information that the muscles are following to know where they go is not given by genes, it's not given even by developmental anlagen, because clearly this digit is the second digit, embryologically and genetically. How do we know that? There's 3 phalanges and so on. But still the muscles go there because they just go by the position. These muscles, they develop and they think, oh, we're going to the sum, but it's not a sum. It has 3 phalanges and etc., but they don't recognize it. Just blind kids going and touching what should be there. That is extremely important if you think about it, because first of all, we have a theory for the connection between muscles, tendons, and bones. Second, it shows clearly the logic of monsters. What does it mean? We can have kids with 4 digits or 3 digits in there, etc., and still we have this thing that happens in real nature, right? With other animals, because these are the constraints. You cannot avoid doing this. The muscles follow the cartilage and where they are, right? And also this decoupling muscle, right? So why is this important? Because even if you lose a digit, you can still have a semi-functional, of course, it depends on articulation and so on, right? But you could have a semi-functional kind of sound, right? You are not in all or nothing, and you will understand better this in the next slide. Because at the same time we were doing these studies, Yuang et al. Did a study with mice, experimental EVO-DEVO study, that showed something that really combined with what we see. When we see these muscles on the forearm, they seem to be a single muscle, but they are not. They have at least 3 different modules. One of them, the tendons form from the digits, not the opposite way. They form here from the cartilage of the digits, and they depend on the cartilage on this region of the autopod of the hand and foot. They develop from the distal to the proximal, amazingly. Then there is a second region or module where the tendon just develop here from the beginning, but is not contacting the other tendons. And then the third region is here where the tendon develop here, as you know, connected to bones, and then the muscle mass develops. In the beginning, these tendons don't connect each other. There is no single structure, but slowly they connect to each other. But what is the importance if you combine these two studies? Well, if you have a human without a thumb, right, you can have this fleshy muscle developing still because you don't even connect. It does not know that there is no thumb there. Then it connects with this tendon, okay, and this tendon here, right, connects to this tendon, and so now you can have a muscle that normally goes to the first digit that now goes to the second digit, because we have the coupling that allows, again, a not all or nothing model. It does a lot of flexibility in evolution. That's what we see in salamanders and frogs and so on. You have this flexibility that you have a kind of a thumb even without a thumb. You have kind of a pinky without a pinky, and so on. Now we are applying this with other colleagues to do surgeries and so on. Why? Because again, we can predict. That's the difference now. Even birth defects can be predictable because they are all these logics, all these patterns, right? So this girl has 8 digits in each limb. Here they were not separated yet, and she has a lot of surgeries in a row. But now we can say to the surgeons, look, we predict that she will have these muscles on that position and these nerves and so on. Because people say, oh, but you can do MRI before. It's not the same precision yet, not the same to see this really small soft tissues and so on. So we predict, of course, we're not 100% right, probably, but you can interact and the surgeon says, oh, this was a bit different and so on. But actually we have already done studies with surgeons in a lot, 54 cases of polydactyly, for instance. And we have seen that in general, when we do these studies, etc., not studies, they are doing surgeries, right? And we predict what they will see and then they report or not. In most cases, yes, almost always our model applies, our topological model. There are some exceptions, and those are amazingly interesting. What are they? Why are the exceptions, right? Why are sometimes exceptions? It seems to be for small things, like if the extra thumb, for instance, is very small, just the size of it won't attract the muscles, and the muscles then go to the other thumb, for instance, not to the most lateral or radial. It depends on things like that, on angles, on size, and really things we never predicted. We were thinking it was the genes or gene expression. No, it's mainly like really small physical things that are controlling. It's really interesting also to see the exceptions. What is interesting is that this model that is being applied in the limbs, going as we say, to the nearest neighbor, right? This nearest neighbor model is very different from the head model. The head model we know is not for nearest neighbor. They don't go, the muscles go to the bones of the same arch. It's a very strict thing that happens in all vertebrates. Probably because the head is globular, it's very complicated if you have a nearest neighbor. Things will be just a mess with all that flexibility. So in the head is like fine and thick muscle, arch 2, fine and thick bone of arch 2. But there is an exception, very interesting. The facial muscles that only appear very recently in mammals, they broke out those constraints and they begin to be again modeled as the limb muscles, which seems to be the ancestral model. Why do we say this? Because invertebrates like flies and so on, they seem to also go by topology, by nearest neighbor. So that seems to be the ancestral model. Then the head changed and got really the fine and thick model much more important for a global complex head with like 100 muscles or something. And then the facial muscles diverted and changed from that model. They are much more flexible, right? If you think about the facial muscles are arch 2, facial innervation, right? A facial nerve, but they can go even to the scalp, the region of the scapula. They can go to the lips, to the skin, things that are not arch 2, right? So it's really interesting to see that. We know also because of birth defects, for instance, people with cyclopia, kids with cyclopia, they don't have a nose, and as you know, it's not genetic; it's epigenetic. Because the height is don't separate, the proboscis cannot go down, and so they don't have a nose, but they still have a nasalis. So the facial muscle is developing where it should be, near navel, as the limb muscles, not as the other head muscles that don't follow this kind of pattern. This is really interesting, actually. About limb regeneration, it's also, again, very interesting to see. As you know, salamanders are really used to study limb regeneration. But my question when I did this study in Germany some years ago was, are they really perfect in regeneration? One thing is to say, oh, they're kind of functional. They move the digits when a new limb and etc. But are they really like a perfect anatomy that regenerates? And so the nerves, the muscles, and so on. So this, as you probably already know, this is actually, they are alive, salamanders, so they don't die and etc. They are just, they have a fluorescent color that can be seen in microscopes. You can compare every single day. And people there, they cut the limbs with anesthetics, etc., and they regenerate them. They don't die, the salamanders, and so on. And so we can see how the muscles regenerate. And I will not talk about that here. After 30 days or so, they have kind of a regenerated limb. But are they really normal or similar to the wild type non-regenerated ones? Actually, they are not. And this is really interesting, mainly because one muscle is really strange. So normally, this is what should happen in a salamander normally, right? The common phenotype. There are only mainly two muscles, not three, as we have in humans. In this region, they have only main two ventral muscles. But in the regenerates, almost 43% of the cases, almost half of the cases, there is like a third muscle, as you can see, the fleshy fibers, not tendon, right? Because there is a tendon here between these two. But here, the tendon is supported by fibers. It's like almost like a third muscle happening as a defect in regeneration, not in normal development. Well, this is amazingly interesting because before that, in our books, we had already said that the tendon that is called coracoradialis, from coracoid to the radius, the tendon of salamanders, just the tendon, not the extra muscle we see in the regenerates, probably gave rise to the biceps of amniotes. And we are seeing in the defects of regeneration exactly what happened in the real evolution from towards amniotes. So this is just like you can see live how this trilogy again, how defects of something in this case of regeneration mirror what really happened in evolution, and probably variations and things like that happens in humans, right? So perhaps in biceps, in some humans with variations, will be only a tendon and not the muscles. That you see is a very interesting correlation. Just to finish, I will give one last example that has to do a lot with these subjects and also let us understand again macroevolution. I don't know if you know, I will not talk about this in detail, but neo-Darwinists were really scared and afraid because Darwin was really exaggerated by neo-Darwinists. But yeah, he did not like jumps in evolution, right? He did not like jumps in evolution, Darwin also. But neo-Darwinists were really paranoid with these jumps. One could not have these jumps. So people like Goldschmidt, when they talk about awful monsters, they were saying, no, this cannot be monsters. Also because a bit of Darwin, right, and neo-Darwinism, especially because they are very adaptationist, everything has to be optimal. So how can you have a macro mutation giving a being that is optimal from day zero? It's not possible. There are no viable optimal monsters, right, with macro mutations and so on. But of course they are. We know they are, right? So what we did with some colleagues like Dinard, we use human names of birth defects and apply them to other animals. Dinosaurs clearly 100 years ago will be recognized as human monsters, or today as kids with huge limb defects like Amelia, zygodactyly, and things like that. You know, their limbs. Why? Because their limbs did not need the upper limbs in many lineages, so they could just vary, and you have huge variation without selection. And so these things, they could be awful monsters because, like, you know, they will not die. They are good enough. They are good enough. You don't need to be optimal to survive. You just need to be good enough. This is good enough for what they needed, right? It's good enough. I will talk about this more, but many books are really changing this paradigm, talking more about awful monsters, and so on. I talk this in my book, so I will just recommend people, if you are interested about this topic, how things are changing, and awful monsters now are getting in vogue, and so on. But the point is to understand that we kind of knew this, like also even before, right? Because we know that all these homeotic mutations are awful monsters. Like in this case, for instance, you have the wing here. In this case, the wing is the second segment. Clearly, this was a macro mutation that just changed where the wing is, first or second. But it's not about optimal. It's just good enough. It doesn't really matter so much if the wing is on the first segment or second segment. It can matter a lot in some lineages, but not so much in others. It's like whatever, it's good enough to survive, right? And we know that these are a result very likely of macro mutations, right? So these are awful monsters, right? The one that is not the ancestral one is an awful monster here, right? So we study especially chameleons. Chameleons are an amazing example of awful monsters because they have things, again, that in humans are considered to be hardcore birth defects. They have syndactyly, so kind of fusion of digits, right? Because they have two super digits, and they have zygodactyly, something I cannot do because it's really messed up, right? But it's that this super digit can go against the other super digit. Now, when those macromutations happened, they happen in humans. We know that macromutations, they happen in humans. When they happen, that jump, we're not saying that from day zero, they were like, oh, I will use this to go to trees. No, they were not optimal to go to trees. They are actually probably very, very much suboptimal for almost all the things after these things happen. But they were good enough to survive. First, good enough. And then after millions of years, they specialize in using these. And actually, they are among the best lizards, the most successful in terms of species and so on, and mechanically to move on trees precisely because they use these kind of defects. It's very important, you know. Evolution is nothing more than defects. Is a defective copy. Right? If not, there is no evolution. So this should be common sense that evolution is just defects. And we are all, in a way, awful monsters because of that. But these are macro-evolutionary cases of monsters. And so we are studying the development of chameleons. We are doing grants and publishing papers and so on about how these things come to be. So to finish this talk, the main topic is so-called logic of monsters. And this is extremely important for medicine. As I showed, you can predict things. Evolution is so constrained that even when things go very badly, or at least not in a normal way, you have seen these patterns repeated, natural variations, and natural variations repeating a normal evolution, and also birth defects. We have all the time repetition of patterns. And this allows us to have predictability in medicine. Those hardcore birth defects are still predictable. They are not a lack of homeostasis. Yeah, they can die inside the mother and so on, but still there is mainly a constraint that cannot be avoided. Some are avoided. That's why, for instance, we have one eye instead of having two in newborns with or fetuses with cyclopia. But many of the other developmental processes are preserved still, right? And this is a very important thing. So this is mainly what brings together this whole studies. And again, the more theoretical and evolutionary, and also some of the medical implications, the broader discussions are in this book. So I think that is explore more these subjects. But here it was just to show you the more specific case studies. So thank you very much. And I hope to see you in the symposium. Again, we'll do short summaries of our talks. Our colleagues complement these with neurobiology, with studies of genes and so on. And so then we have a roundtable to really address this perhaps more holistic way of also see a more macro way of seeing evolutionary medicine. So thank you very much.